The Bap1 tumor suppressor gene plays an important role in mesothelioma development and progression. Mutations in Bap1 occur in approximately 50% of malignant pleural mesothelioma cases. Bap1 loss promotes mesothelioma growth and reduces sensitivity to chemotherapy. As a result, clinical trials are actively investigating agents specifically targeting the Bap1 pathway or exploiting Bap1 defects in mesothelioma tumors. One of the first clinical trials to select patients based on their Bap1 status is a phase I study of the PARP inhibitor veliparib in advanced solid tumors including mesothelioma, NCT02286826. Poly-ADP ribose polymerase, PARP, inhibitors work by trapping PARP at sites of DNA damage, which leads to replication fork collapse and ultimately cell death, specifically in cells deficient in homologous recombination repair pathways such as Bap1 mutated cells. Preclinical studies showed veliparib suppressed growth of Bap1 mutant mesothelioma cell lines and tumor xenografts. The goal of this trial is to evaluate the maximum tolerated dose, safety, and efficacy of veliparib monotherapy specifically in Bap1 mutant mesothelioma patients. Correlative studies will also assess PARP trapping in circulating tumor cells and tumor biopsies. Other PARP inhibitors are also being studied selectively in Bap1 mutant mesothelioma. A phase I/II trial is planned to investigate the PARP inhibitor niraparib in Bap1 deficient mesothelioma and triple-negative breast cancer patients, NCT03896780. Preclinical data demonstrated synergy between niraparib and inhibitors of WEE1 or CHK1, two kinases involved in DNA damage response. Based on these findings, the trial will also explore the safety and efficacy of niraparib combined with the WEE1 inhibitor AZD1775. Correlative studies will profile molecular alterations and response biomarkers to PARP inhibition based on tumor Bap1, BRCA1-2, and homologous recombination status. One approach to exploiting Bap1 loss involves reactivating the MTOR pathway, which is suppressed due to loss of Bap1 tumor suppressor function. A phase I/II trial evaluated the MTOR inhibitor temsirolimus in Bap1 deficient mesothelioma and other solid tumors, NCT02393794. Preliminary results showed temsirolimus achieved durable stable disease exceeding 6 months in 3 out of 8 Bap1 mutated mesothelioma patients, supporting further investigation of this strategy. A separate phase I/II study is examining the safety, tolerability and preliminary efficacy of combining temsirolimus with the EZH2 inhibitor tazemetostat specifically in Bap1 and or SET2 altered mesothelioma, NCT04176199. EZH2 suppresses target genes of growth inhibitory pathways and its inhibition may augment temsirolimus activity in Bap1 deficient tumors. Additional strategies targeting downstream effects of Bap1 loss are also in development. Since Bap1 loss leads to elevated levels of hypoxia-inducible factors HIF1A and HIF2A in mesothelioma, a phase I/II study is assessing the safety and efficacy of combining the HIF2A inhibitor PT2385 with standard chemotherapy in untreated mesothelioma, NCT02655822. An expansion cohort will specifically enroll Bap1 mutated patients. Preliminary results identified a potential association between Bap1 mutations and improved progression-free survival on the PT2385 combination, providing rationale to further assess this strategy selectively in Bap1 deficient tumors. Finally, immunotherapies may offer another approach to target Bap1 mutated mesothelioma based on preclinical data showing Bap1 loss enhances tumor immunogenicity and sensitivity to PD-1 blockade. A phase I/II study is actively recruiting Bap1 mutated unresectable malignant pleural mesothelioma patients to receive the PD-1 inhibitor nivolumab alone or combined with the CTLA-4 antibody ipilimumab. NCT03311461. The primary objectives are to characterize safety and identify preliminary efficacy signals of these immunotherapy regimens selectively in the Bap1 mutated mesothelioma population. Early results have shown promising objective responses that appear durable in a subset of Bap1 deficient mesothelioma patients, supporting further investigation. Clinical trials are specifically targeting the Bap1 pathway or molecular consequences of Bap1 loss by exploiting synthetic lethality with PARP inhibitors, modulating MTOR activation, inhibiting HIF downstream targets, or assessing immunotherapy efficacy based on preclinical rationale. Correlative biomarker studies will help determine which strategies show the most promise to selectively benefit Bap1 mutated mesothelioma patients. Further investigations are still needed, but this progress highlights how identifying actionable gene mutations in cancer is helping guide precision, biomarker-driven clinical trial design.